So I guess my research, I like to kind of work on developing Bayesian methods and applying them to different kind of applied problems, so particularly like societal challenges, things like health and social sciences applications. And so today I'll talk about work that I did at my last postdoc at the University of Warwick in the Neglected Tropical Diseases Modelling Consortium, and also some work that I did in the PhD looking at COVID applications because they were the most relevant for this audience, I guess. Okay, so I guess the title of the talk is this, but I kind of want to talk about it in terms of these four interconnected kind of themes. So the first is looking at how we can use Bayesian statistics to understand things in the world around us. The second is looking at actually developing methods to help us and allow us to do this. And then I guess because I'm quite on the applied side, I like to make sure that things are accessible so that people who aren't statisticians can actually get use out of these things. And maybe the most importantly is trying where possible to have this kind of interdisciplinary approach to research. So working directly with the people that you're trying to help and make these tools accessible too. I mean, I will probably go through this quite quickly because I'm assuming we all know what Bayesian statistics is. There are some reasons why I like to use them, particularly for this like interdisciplinary research. I think there are a lot of useful aspects of it. But we can't always compute our model evidence, the denominator. So if we step back, what might we want to use these methods for? We have our parameters theta, data y, and pretty much everything we want to do related to theta comes down to some kind of expectation, which is this like potentially high dimensional integral. And we know through Monte Carlo integration that if we can sample from our posterior distribution, we can then plug in those samples into whatever function we're interested in, take the mean, and compute things like means or uncertainties or probabilities. And so if we can do that, that's great. We can work with our posteriors and get all of the different kind of inferences that we want, but we can't always sample from theta. And so this is where a lot of Bayesian methods come in. So the ones that I'll talk about today are importance sampling and MCMC. I guess importance sampling, you have a distribution that's hard to sample from, so you instead sample from an easier distribution and then correct for the fact that you're sampling from the wrong one. MCMC, you want to construct a Markov chain where the linear distribution is your posterior P of theta given y. So I guess I think Bayesian tools are really nice to
just let me see if I can get this this window out of the way.
Oh, yeah, sure. Yeah, so like I mentioned, I'll talk about two kind of applications of Bayesian statistics for applied problems today. The first is looking at controlling neglected tropical diseases, and the second is around modelling some kind of aspects of COVID transmission. There's kind of another branch of work that I've done looking at conflict and political violence events, which I think is kind of like a nice application of these Bayesian methods, but not really relevant for this audience, I think. So if anyone happens to be interested in conflict, feel free to come and chat. I'm very happy to talk about it. But yeah, I guess today I'll focus on these other two branches. So first looking at informing policy for neglected tropical disease. So I guess I assume many people haven't heard about them. It's in the name, they're neglected, they don't get a lot of attention. But they do impact, I guess, more than 1 billion people worldwide. There's quite a high burden to these diseases. And if we look at deaths and kind of disability adjusted life years, there is these can be quite debilitating, but they receive a lot less attention than other things like tuberculosis or malaria. And so I guess as a result of this, they get quite limited resources to actually control the disease of these, control the spread of these diseases. There are kind of initiatives in place to try and reduce the control of these diseases. So getting rid of neglected tropical diseases is part of the kind of SDG 3 around good health and well-being. There's also the WHO kind of roadmap towards ending neglected tropical diseases. So there are kind of these frameworks in place, but there are still quite limited resources available to actually tackle these things. And so I guess the NTD Modelling Consortium, we're trying to answer questions around how can we best allocate these resources, given that they are. And so yeah, the NTD Modelling Consortium, it's funded by the Gates Foundation. And so it really brings together kind of data scientists, statisticians, epidemiologists, geostatistical modelers, to answer kind of urgent policy questions around how we should be controlling NTDs. So these are things like, where should we be treating? How often should we be treating? Kinds of questions. So I guess it's Gates funded, so our main stakeholder was kind of the Gates Foundation, but I guess other kind of key people who are interested in this work are like the WHO and various kind of national programs. So these are the NCD programs actually in the countries that we're working with on the ground, kind of carrying out the treatments and these kinds of things. Yeah, so the NCD Consortium, we were funded to work on 8 tool-ready diseases. So these are diseases where there are safe drugs that you can just go out into a community, treat everyone, regardless of whether they're infected or not. And you do this kind of at repeated intervals. And this is quite an effective measure of reducing the prevalence of these diseases. So yeah, the 8 diseases that we focused on are on the right here. So trachoma is the only bacterial infection that we had. It's a bacterial infection of the eye. So in more advanced cases, it causes like scarring on the eyelids, which then causes blindness. The next group of 3, they are STH or soil transmitted helminths. So in the name, they're transmitted through kind of contaminated soil. The next group of 2 is schistosomiasis, and they're transmitted through snails who release the kind of parasite into water, and the water then comes into contact with people through their skin and infects them in that way. And these kind of 5, so the STH and the schistosomiasis diseases, if you have quite a low infection, low burden, meaning maybe you only have a few worms living in you, the symptoms generally aren't too bad. The main issue comes when you have kind of repeated chronic infections. So at more advanced stages, it causes things like gastrointestinal issues, so like abdominal pain, diarrhea, vomiting. And in children, it can cause like malnutrition and stunted growth. So if you keep getting infected by these things, then the kind of burden becomes quite high. Onchocerciasis is the next disease. It's transmitted through black fly bites. And it's also known as river blindness. So it's one of the leading causes of kind of blindness and skin disease in Africa and the Americas. And lastly, lymphatic filariasis, which is another parasitic worm that's transmitted through mosquito bites. And it can cause things like the kind of really severe swelling that we saw in this slide. So obviously quite a high burden. It's, it impacts the people who are suffering from this quite a lot, but there's also a lot of kind of social stigma around it as well. So yeah, they're the 8 diseases that we were focusing on. And what we really wanted to know was at a kind of fine spatial scale, what's the disease prevalence in these areas? And what are the kind of factors driving transmission at a fine spatial scale. And so I just go through like the pipeline of the kind of framework that we're working with in the consortium. So we take a geostatistical map of disease prevalence and combine this with some kind of transmission model that tells us how the disease is spread. And we combine these things to get kind of fine scale estimates of the model parameters. So this is just an example of like Ethiopia for one of these diseases. So once we have these kind of subnational estimates of the model parameters, we then feed these into projections of prevalence in the future. So the Gates Foundation or kind of other stakeholders might be interested in different treatment scenarios. And so they say, if we carry out treatment scenario 1, how will prevalence look like by 2040 compared to these other treatment scenarios that we're interested in? So we do that, and then that feeds into things like health economics and cost effectiveness analyses, and then it all feeds into kind of inform how we should be controlling these diseases using this kind of data-driven approach. So just to kind of go into the different aspects of these in a bit more detail, I only really touch on like this first half of the pipeline because that's what I was most involved in. So we worked with geostatistical modelers, like I said earlier. So they developed a kind of Bayesian geostatistical model of prevalence. So, for example, if we had different kind of survey locations in the black crosses here, we then take that and fit some kind of geostatistical model to get like a surface of the prevalence. And it's a Bayesian model, so we would take samples from this geostatistical model. So we'd essentially have, if we wanted 1,000 samples, we have 1,000 of these surfaces. And we then feed that into our model as kind of data. So our data is technically like a posterior from a geostatistical model. If that makes sense. So essentially for each kind of pixel here, we would have 1,000 draws from the posterior of what the prevalence might be at that location. I should say this is just an example of Ascaris, which is one of the parasites that I talked about earlier. So the next part is the transmission model. So it can be any type of model you want. It can be an agent-based model or a deterministic function, really anything that tells you, given these parameters theta, what is like the prevalence that we would simulate from the model. And the thetas that we're interested in are really those ones that are kind of related to how resilient to treatment the parasite is, or different kind of mechanisms that drive that resilience to treatment. And yeah, so we want to estimate these parameters and then draw from that some kind of H theta. In this case, this is a prevalence resulting from a transmission model simulation. And so for our Ascaris example, one parameter we might be interested in is, we call it K. So it's a heterogeneity of infection. So it's saying among a population, what's the kind of variation in how susceptible these people are to being infected. And this is just one example of what a mathematical model or a transmission model might capture. So going from kind of within the human host and then through to different kind of vectors that might carry the diseases. So just explaining the kind of life cycle of the transmission. And through this, we would be able to pick out kind of a prevalence or a potential prevalence given the thetas that we've fed into that model.
What data do you have for this beyond the, beyond the prevalence? You must have other data to inform.
Yeah, I guess these models tend to, there's loads of parameters that are in the models, and most of them are kind of preset like they're just fixed values based on kind of previous studies that have been done. And yeah, we only vary the particular parameters that are kind of interested that we know drive transmission. But yeah, there's a lot of, I guess, uncertainty just in the transmission models because they are relying on these kinds of fixed preset values of a lot of these parameters. Yeah, it gets a bit unwieldy because these models can have kind of hundreds of different parameters feeding into them, so you don't really want to be working on that high dimensional space.
May I ask, like, what is the reason for doing these two analyses in two steps, like the estimation of the prevalence and the estimation of the theta?
Well, actually, it's not really two steps, I guess. The transmission model is literally just like we're drawing simulations from the transmission model given some values of theta. So it's kind of, I guess the prevalence that we get out of this is kind of like our data that we're then feeding into the posterior. If that makes sense. Yeah, so I guess the next step is that we take kind of the geostatistical map samples, which is technically a posterior distribution, but we're just pretending that it's data. Then we take our transmission model and we then generate these kind of subnational parameter estimates. But these transmission models are quite expensive in a lot of cases. So we don't want to, like for each of these different subnational regions, we don't want to be drawing, you know, hundreds of thousands of simulations for each of the regions. So we want a way to estimate for each of the regions our parameter, for example, like K, but not have to run as many simulations. And so we're using adaptive multiple importance sampling. So it's just an important sampling scheme that is like recycling the samples that you've drawn at previous iterations rather than kind of throwing them away like you would normally do. So if you go break down the different components of this, we have, you know, important sampling. We've talked about that already. Adaptive is named because we're updating the proposal at each iteration. And multiple is because at each iteration, we're kind of generating the proposal by using all of the samples from the previous iterations to kind of inform where like a good area of the kind of posterior or the values of theta could be. And so we developed a kind of newer version of this AMOS algorithm for this work. Which I don't really talk in like a lot of detail about it, but I just want to give like a flavor of what we're doing. So we're essentially, if we look up on the right here, this is one example for just a single location. So each of these black lines is a simulation from the transmission model associated with a particular value of theta. And the red violins are our distributions from the geostatistical maps. So these are kind of the distribution of prevalence at a particular location based on our geostatistical data. And so in the first iteration, we're literally just drawing from the prior a bunch of values of theta. We then feed that into the transmission model. It gives us the different prevalences throughout time. And then we will weight each of these simulations according to how closely they line up with our geostatistical map samples. And then we're using an adaptive algorithm. So at each iteration, we'll then kind of use those weights to inform where we should be kind of proposing the next round of theta values from. And so we just repeat this process until we reach some kind of effective sample size threshold that we set in advance. And so at the end of the day, we end up with a weighted sample of values of theta. And we can then kind of subsample from that weighted set to get, say, 200 values of theta that give us kind of reasonable simulations of the prevalence values that fit through the map samples. So we're basically doing this for kind of every subnational region that we're interested in. Does that make sense? Yeah, so I mean, this is just kind of a summary of what we, during my time at the consortium, we did to this AMOS algorithm. So a likelihood function in our posterior, we actually don't need a likelihood function. We tend to just estimate it kind of empirically. And yeah, we did some improvements to improve the stability of that. We also asked to estimate the model evidence from the kind of weighted sample that we have. And we also estimate in some cases the secular trend, which is kind of this idea that if you improve people's access to kind of clean drinking water and improve hygiene and sanitation, that will naturally lead to a decrease in these diseases. Because these diseases are only really found in places where this isn't the case. So yeah, you can kind of capture some declines in prevalence just through this kind of secular trend. And we have an R package available if anyone wanted to, has, you know, their own data and transmission models that they want to plug in. So yeah, they are the kind of updates that we did to the algorithm. But overall, for all of the 8 diseases, we were looking at different kind of epidemiological parameters, ends up being over like 5,000 regions across sub-Saharan Africa. And so we're able to obtain these different model parameters for each of these regions. I don't really go into all of the kind of results, but this is just an example of what we might see. So for this K parameter, this heterogeneity that we spoke about earlier, if we look at our map of the kind of means from the posterior, we can see regions with kind of higher K, regions with lower K. And a higher K suggests that the infection is more homogeneous across the population. So maybe people tend to have a more similar number of worms that they're infected with. Whereas if you have a lower K, very low values of K are kind of what sustain prevalence. Even if most people aren't infected, you still have some people that are just really, really highly infected. So no matter how much you decrease prevalence in the majority of the population, you'll still have those like really highly infected people that keep kind of the disease spreading. And so this is why you see this kind of relationship with, we take one of the prevalences, so in year 2000, those areas with a higher K have high prevalence, and those with a lower K and have lower prevalence, because most people aren't infected, but you have these few people that just kind of keep the spread going, in a sense. And so these are just the means, but we can also kind of get a whole distribution around the primary. Okay. Yeah, we did this for all of the diseases. All of the diseases, there were slightly different parameters that we were interested in estimating. And I can't really show like the actual projections, but I just kind of give an example of what we might be looking at. So we then take these subnational estimates and we have some kind of treatment scenarios. So in this example, say this yellow scenario is, I don't know, say we don't do anything and we just keep going as we currently are. What will transmission look like in 2040 compared to alternative scenarios where we might be looking at things like treating more often or increasing the coverage of how many people we're treating at a time, maybe like a hypothetical new drug or vaccine. Yeah, we can kind of do these projections and see if we went with this scenario 1, we can see that we're not really doing much by the way of reducing prevalence, as opposed to these kind of more intensive scenarios. Yeah, so it's kind of a very brief summary of what I was doing at the NCD Model Consortium. So we developed these extensions to the AMOS algorithm that allow us to get these subnational estimates of model parameters. And really the end goal is to kind of guide the treatment strategies of these diseases and inform how we should be kind of trying to control them. And we also have an R package. Yeah, okay, that's all I wanted to say on that. Any questions before I move on?
You mentioned that one of the advantages of AMIS is that you are able to estimate the marginal likelihood, the model evidence. That's definitely a nice thing to have. Are you using that in this example? Are there multiple models that you're comparing? Or is it just that, I mean, it's in general a nice thing to have if you want to do that?
Yeah, I mean, like the from what I've talked about here, no, but there are kind of, I guess, side quests that we were going on that we did want to actually compare different types of models. Yeah, I guess in like the main body of work, we weren't really using it, but there are kind of certain questions that we might have that it was useful for.
Thank you.
Because there's kind of different versions of some of the transmission models as well. So we can run both of them and then calculate them.
It was the same question.
Okay, cool. Right. So the next thing I'll go through is a kind of different way of looking at COVID-19 transmission. So I guess I don't need to hammer on about why we might care about modeling COVID transmission. But I guess the main point of this body of work is to kind of provide a way that we can model the interaction between different countries or regions. And was spreading throughout the first part of the pandemic. So we're proposing Hawkes processes to model these interactions, multivariate Hawkes processes. Have we heard about? Okay. Okay. Yeah, so in this case, we're taking as an example just Italy and France. And our observation window is basically the start of the pandemic through to May 2021. This was when I was working on it. So that was all the data we had at the time. And I mean, we can see if we look at the data, there are these kind of different phases. So the first is this kind of explosive phase of a kind of exponential growth, which we call phase 1. And then we have this downwards phase where kind of the non-pharmaceutical interventions and all of these lockdowns and everything is happening. So we call that phase 2. We then have phase 3 being the kind of start of the second wave, and then phase 4 is kind of decline after that. So we've just kind of manually identified these change points in this way. And so a Hawkes process is a self-exciting stochastic process. So we can basically use them to describe any phenomena where one event happening increases the probability of future events happening. Whereas if you have something like a Poisson process, your events are kind of happening independently of one another. And in this case, we're using discrete time Hawkes processes because traditionally it's a continuous time process, but we have COVID counts of, in our case, we're using deaths rather than the cases, but they're aggregated counts. So we don't know when each of these kind of events happen. So we're just kind of doing a discrete approximation of the continuous time Hawkes process. And so if we define our Hawkes process as NT, it's literally just keeping track of how many events have occurred up until time t. And then we have YP, which we call the kind of size of each of the jumps in that process. So if we have NT on the y-axis here, up until time t1, we have no events. Then it jumps up by YT until time t2, jumps up by YT2, and continues in that way. And so in our case, we're assuming that the size of these jumps, YT, is Poisson distributed with conditional intensity function lambda, where lambda is conditional on the events that have happened in the past. And so if we have YT as Poisson with great lambda, and this lambda is basically the expectation of the size of those jumps. And we break it up into two kind of processes. So the first is the kind of baseline process. So this is the rate of events that we would expect just to occur independently of each other. So if you had a Poisson process and ignored this kind of second term, that's what we have. We then have this self-exciting process, which is telling us, well, yeah.
I don't really understand. Talk about it being discrete time, but those times T1, T2, T3, they seem to be different distances apart.
Oh yeah, okay.
So is there some process that's determining when T1 is and when T2
No, this
is?
is just like I mean, you might have like multiple integers. This might be like T1 might be 5, so you didn't have any events occur up until time 5, and then it's going to be like time 8, for example. Like all of these T's are integer values.
So I should regard the T1, the T2 and so on as being equally spaced?
Not necessarily, like there might be, they're not, I guess there might be like various integers in between them, but I just haven't. I guess this is just kind of an example. Demonstrate how.
So is Y, you got Y subscript little t. If T1 is 5, then are Y1, Y2, Y3 and Y4, are they all equal to 0?
And yes, well, I mean, they still have some kind of rate associated with them, but the actual observed counts will be 0.
Okay, I think I understand now.
And so each of these, this subscript T is an integer.
Okay.
So yeah, we have this self-exciting term, which is telling us for each of this kind of groups of events that have happened in the past, what's the kind of additional contribution to the intensity function that we would expect. So if we break down the different terms, alpha is a magnitude parameter. In the context of COVID, it's like something like an R0, but not quite. And then we have a sum over all of the previous events that have happened. So S being the kind of indicator of, so say if we were at time T3, S would really only need to be T1 and T2 because they're the only times that events have happened in the past. And YS is just this number of events that happened at each of those points. And G is kind of the key feature, I guess, of the self-exciting process. So it tells us, given how much time has passed since those events occurred, what does the contribution to the intensity do? And if we plotted lambda, we would then see something like this. So we have mu up until time T1 before any events have occurred. The rate is just kind of baseline rate. It then jumps up after we have events occur and then decays at some rate until T2, where the second event occurred. That spikes again, then decays and keeps going. And so just to show like an example of what this G might look like. So we have the value of G on the y-axis, and on the x we have the time that's passed since a particular event occurred. And so in this example, we can see like immediately after an event, we have the highest kind of contribution to the intensity function, and then that then decays as we move to the subsequent. So this is really what kind of controls the shape of this decay. And yeah, in the context of kind of what I was talking about at the very start, so we have theta, the parameters that we're interested in estimating are kind of our mu's, alphas, and whatever parameter this decay kernel. And H, we might be interested in calculating like the rate of events, because this tells us kind of the expected number of events that we would, the expected number of events to occur at time t. And in this case, we're looking at a multivariate setting, so we want to consider France and Italy kind of two separate Hawkes processes that are interacting with each other. And so this is all expressed in terms of like k dimensions, but in our case, we just have two. So we have k kind of Hawkes processes that are interacting with each other, and in red are the kind of additional components that arise as a result of this multivariate set. So each of our Hawkes processes now has its own corresponding YTK, which keeps track of in the number of events in that country. And that's also indexed by a separate lambda function. Our baseline rate is also defined by K, so both of the countries have different levels of baseline activity. And I guess the key part is that our self-exciting process is now a sum over all of the events that have happened in both of those, in our case, dimensions. So we're interested, we get kind of additional contributions to the intensity from events that happen within a country, but also in the other. And they're associated. And one more thing is that because we have these different phases that we've defined, we have kind of a separate multivariate Hawkes process occurring in each of these phases. Just because the way the Hawkes process works, it doesn't really make a lot of sense if you're changing the kind of dynamics this severely. So we thought it was kind of simpler to just consider them as separate. So
And you define those phases post hoc, given the data, not according to policy change dates or anything.
yeah, I mean, this was like, we were kind of doing this right at the start of the pandemic. So I guess we were prioritizing simpler methods just because we wanted to kind of just see how Hawkes processes worked. We were like, would they work in this case? Yeah, there are definitely like ways that you could improve that, but I guess in the multivariate setting, it's also a little tricky because the peaks are happening at slightly different times in the countries, and so it's not like super trivial.
Did you set sort of orders of 3 phases? Did you set them to one date, or did you introduce some?
Yeah, no, it's literally a hard.
Do they share some information, the priors and these parameters, like between the 4 models that you have? Or
I guess they're sharing data in the sense that, like phase 2, we're still including all of the events that happened in phase 1 in the data, but they have the same prior as well on them. We didn't set like phase specific priors.
And the phases, they are shared across dimensions, right? So, because here it looks like, for example, phase 1 looks like it makes more sense for France than it does for Italy.
Yeah. Yeah, I mean, yeah, we could definitely improve maybe the way we're defining the phases.
So were they determined by looking at one of the countries and then fixing those and then?
Yeah, I mean, it was quite ad hoc. Like we looked at the data and said, this looks like a change point. Yeah, because I guess like I said, it's quite non-trivial to have a more automated way of doing this, just because yeah, the way the whole process is a lot of its kind of stability conditions are dependent on whether it's like an explosive or a non-explosive dynamic, and so it's
also, as you were saying, we should bear in mind the context in which you were doing this as this was happening. Right? So you were inventing the stuff as it was required. So yeah.
Okay. And so I guess the main point of this part of the talk is we were trying to find different ways of defining this, these triggering kernels, or in this case, the interaction kernels. So they're telling us how. And so, because generally we would just use some kind of parametric form, like a exponential or a geometric in the discrete case. But if we do have more complex interactions happening, obviously this isn't really going to be sufficient. And so we thought a more flexible way of doing this is like a random histogram kernel. So basically we set our histogram so that these interaction functions can basically take the form of any step function. So we have, in this case, is an example of a 3-step or a 3-component histogram. So we have the various heights of the histogram and different change points for which these steps occur. But I guess the thing is that the number of components in our model is unknown, so it can be up to, I mean, we impose some kind of maximum excitation time. But so, yeah, the shape of this kernel can be any step function with that many components. And so these heights and where the change points occur is also random. So we're using reversible jump MCMC to estimate this with birth-death moves. So if we start with our kind of 3-component histogram and say we want to propose a birth move, we would then sample from kind of the set of vacant change points. Say we choose this S star, we then simulate it or generate a kind of a corresponding height for that component. And that would be a 4-component histogram, which we then accept or reject with some probability. And then if we want to propose a death move instead, if we then take our 4 components, say we, from the set of current change points, we choose S star, we then remove S star and the corresponding height to then regain 3-component histogram, which we then accept or reject with some probability. We also tried like a split merge move, but that didn't really do much and it added a lot of kind of computation. So we, yeah, so just to kind of end, this is an example of what we got from this model. So for each of our phases, we have a different group of kind of interaction functions that we estimated. And so on the diagonal are the kind of within the country interactions. So how is the kind of contribution to the intensity function changing based on events that occur within a country? And then we, on the off diagonal, we have the kind of cross excitation patterns. So in this example, this is saying how do cases in Italy impact the intensity of cases in France and vice versa. So I guess the main thing that we thought was cool out of this work is in phase 1, we can see this kind of Italy to France interaction. So Italy was one of like the first countries that had kind of widespread infection. And so we thought it was kind of cool that France to Italy didn't really show much excitation, but Italy to France, we can see this kind of 10-day delay before this kind of peak in excitation. So, you know, people being infected in Italy coming into France and then infecting there. So, I mean, we thought it was kind of cool that we could capture that. And then as the pandemic continues, we kind of see just within country interaction and not so much like cross excitation. And yeah, I guess it's cool that we can do this, but it's also quite computationally intensive. So I think in further works, you would kind of want to figure out whether you expect there to be like these more complex interactions, because otherwise something like this could have just been captured with like a geometric kernel. So I mean, definitely like avenues that could improve the computational burden of this, but generally, like if you do want to capture more complex patterns, and that's what we can do. So we've talked about these 2 examples. If you want to hear about the third one, feel free to get in touch. But I guess just generally using Bayesian methods for applied problems is computationally is kind of the biggest hurdle that we need to overcome, especially to make them accessible, because people, practitioners, they don't want to be waiting days for their MCMC to converge. And so I think kind of these methods that are focusing more on optimization rather than sampling are quite interesting in the Bayesian space. So I think it's a cool avenue for the future. yeah, so like I said, in terms of these 4 themes, we have understanding, parameter inference, we can just chuck things into a neural network and it'll tell us anything we want. But I think there's still a place for a good old-fashioned parameter inference where there's a specific aspect of some phenomenon that we're interested in. Yeah, kind of leveraging more of these modern data science optimization-based approach, and accompanying this with software and also collaborating with stakeholders, I guess, is also really important. And that's all. I've worked with lots of lovely people throughout this work. Yeah, happy to take any questions.